6:30. It's about time to start. What's going to happen is I'm going to try and spend about an hour on the multiple choice and then about 45 minutes on the written responses. We're going to end about 15 minutes early for you guys to fill out a survey as well as to ask any questions you're not clear on. And we're going to also need to clean up the room in that time. So everybody good? Okay, awesome. So for number 1, what is not a method of control of enzyme activity? C, okay, yeah, C is the answer. Oop, that doesn't work. There we go. So what are some examples of covalent modifications of enzymes? It's going to be A. It's phosphorylating the hydroxyl groups of serine, threonine, and tyrosine. And what is an example of an allosteric effector? Okay, A is 1. Or wait, no. A is not 1? Yeah, A is not 1. C and D. It's going to be C and D, so the answer is going to be E. I don't want to give you the wrong definition, but it changes by binding covalently.
That's an allosteric effector.
Yeah. Binding covalently, just go ahead and look in your textbook and stuff just to make sure I'm giving you guys the right definition. Negative feedback regulation is an example of what? What is negative feedback regulation? So negative feedback regulation is when the product goes back to the beginning of the process and stops it from happening, like stops enzymes from working. It's going to be allosteric regulation. Why would you couple an endergonic and an exergonic process? Oh, pull up. Sorry. It's going to be used to drive the reaction forward. Okay. Does anybody know why it would drive the reaction forward? Exactly. Good job. Okay. Yeah, if you have, usually you pair a cellular process with the hydrolysis of ATP, which releases a lot of energy. So you can pair that with a very endergonic reaction to drive the reaction forward. Yeah, it won't be favorable if you pair a very large endergonic reaction with a very small exergonic reaction. But normally in our cells, we pair it with ATP hydrolysis, which is a very, very exergonic reaction. So, to drive an exergonic reaction forward, what is an exergonic reaction? Okay, yeah. It would drive an endergonic reaction forward. So, pyrimidine is joined covalently to the pentose sugar through the what? A. I have C. What's up?
1 and 2, what were the answers?
1 and 2, 1 was C and 2 was A. So a pyrimidine is joined covalently to the pentose sugar through the blank. I have this one listed as C. Let me go to page 46 real quick. C. And let me try and find this for you guys. Yeah, it's not going to fit under the dot cam, so where you can find that is on page 46, figure 3.3. C. That's the one that I have. So, in DNA sequencing by the Sanger method, C? Are you sure? What is the Sanger method? The Sanger method is the dideoxy method. So, which ones have dideoxy in them? It's going to be A. Dideoxy-CTP is used to terminate the enzymatic synthesis of DNA where Gs occur in the template strand, because in the dideoxy method you are transcribing the template strand, so you're getting the complementary strand. So wherever you have a G in the template, you'll end up with a C. Does that make sense? Yeah. What method would be C? What method would be C? My mind's drawing a blank right now. I'm really sorry. I think it might be Edmund, but I think Edmund does only one at a time. So yeah, it uses gel electrophoresis, so it separates the DNA. I'm just not sure. What? PCR? It might be PCR. I don't really know, though. Sorry. Okay. The size of the DNA region specifically recognized by type 2 restriction enzymes is typically what size? A. I like the sound of A. Does everybody like the sound of A? Okay. So are type 2 restriction enzymes the most selective, or are they the least selective? Most selective, yeah. So what are the other two types of restriction enzymes? I heard someone say type 1 and type 3. Good job, guys. So for the helix and double-stranded B-form DNA, most of the stability can be attributed to what? D? I heard some Ds. Why do you guys think it's D? Okay, it is D. Yeah. Does anybody have any questions on that, or can I move on? This is specific to B form. H bonding is usually not considered super important in B form. Like, it is important, but it's not one of the most important. So base stacking and van der Waals are the most important for B form. And Z and the other form of DNA, I forgot what it was, they have different ones. So the PCR reaction mixture does not include what? There's C, DNA ligase. You guys like C? Yeah, okay, I like C too. So it will be C. And then, does anybody have any questions about that? It's directly on Hashim's PowerPoint, and you can probably find it in the book too. So, when two carbohydrates are epimers, what does that mean? B, okay. It is B. They differ in configuration only around one carbon atom. Oh, geez. This. So, number 12. Which of the following are anomers? It's going to be D, alpha D-glucose and beta D-glucose, because they differ around the anomeric carbon when they're in cyclic form. Okay. Which of the following statements concerning the tautomeric form of bases such as uracil are correct? Move it up, sorry. C? It is C. At neutral pH, the lactam form predominates. Okay. In nucleotides and nucleic acids, syn and anti-conformations relate to what? It's rotation about the sugar-base bond. Because you can rotate it, and syn and anti relate to that. So, number 15. Which statement accurately describes the interactions between the molecules thrombin, antithrombin, and heparin sulfate? Did I have any answers? C? Yeah, it's going to be C. In the presence of heparin sulfate, antithrombin binds tightly to thrombin, inhibiting blood coagulation. I believe in the text it just talks about heparin sulfate being an anti-blood coagulate. So kind of have to mix and match there. Number 16, which glycosaminoglycan is the most heterogeneous? It's going to be keratin sulfate. Keratin sulfate has a lot of sulfur in it. It's got a lot of sulfur in it. It's mostly sulfur, so that's why it's the most heterogeneous. Or, I think that's why. Oof. Okay. Oh, that one's a duplicate. Everybody should know that one already. It's C. Protein tyrosine kinases do what? I'm hearing, I thought I heard an answer. Did you say one? No? Okay. So, protein tyrosine kinases catalyze ATP-dependent phosphorylation of tyrosine side chains, which then mediate protein-protein interactions. So it's going to be A. All of the following is true about receptor tyrosine kinases except what? Yeah, it is going to be C. Why is it going to be C? It autophosphorylates tyrosine residues, not, I think, glutamic acid residues is what I changed it to. Which of the following statements concerning signal transduction by the insulin receptor is not correct? It's going to be A. The substrates of the receptor protein kinase activity are mainly proteins that regulate transcription. So, hormone-activated phospholipase C can convert, oh geez, can't pronounce that, phosphatidyllinous? Don't know. 4,5-bisphosphate to what? It's going to convert it to DAG and what? It will be A. So it converts it to diacylglycerol and inositol? I don't know. Phosphate? So pKa is what? B? Yeah, exactly. It's allosterically activated by cyclic AMP. A ligand-gated ion channel is what? It's D, a membrane protein that permits an ion to pass through the membrane only when opened by the appropriate ligand. If it's ligand-gated, it needs a ligand to bind in order to open. Got it? Which one? D. Glucose transport into erythrocytes is an example of what? It's going to, B? Yeah, it's facilitated diffusion. Which of the following statements about facilitated diffusion across a membrane is true? Have you guys had time to read through? I heard a D somewhere. It is D. A specific membrane protein lowers the activation energy for movement of solute through the membrane. 26. The specificity of the potassium ion channel for potassium over sodium is mainly the result of what? Any ideas? Yes, no, maybe so? A? Yeah, it's A. The differential interaction with the selectivity filter protein. You mix equal volumes of 60 millimolar Na2HPO4 and 0.51 millimolar Na3PO4. Which of the following best describes the resulting solution? It'll be D. The pH will be 12.27 and it'll be well buffered. I don't really do math for these, but I can try. So you see how this is at a different protonation state than this one? Let's see. So this one has one hydrogen left, and this one has no hydrogens left. So you have, which two pKas are interacting together? I kind of already pointed them out. Yes. So Which one has the highest concentration? The first one. But you mix equal volumes of that. So the only thing that really falls in between the two is going to be pH of 12.27, and it'll be well buffered. So, does that help people with those? Okay. The reaction is spontaneous only when T is greater than delta H over delta S. What does this refer to? Gibbs free energy, yeah. So, which one is it? I believe Hashim in her PowerPoint has like a little table of them. So, it's going to be A. Given its mass in kilodaltons, which of the following proteins will elute last from a gel filtration chromatography column? What do you know about gel filtration chromatography? What moves fastest in a gel filtration chromatography? Smallest? Are you sure it's the smallest, or are you getting confused with gel electrophoresis? Yeah, the answer is A. For gel filtration chromatography, the smallest actually moves the slowest because the matrix that you put in the gel captures the small molecules, basically. It makes them go a very, very long route, while the bigger molecules can just kind of push it away. Everybody understand? Okay, awesome. STS page is used to blank and blank based on blank. It's A, yeah. STS page is used to denature proteins and separate them based on molecular weight. Yay! Which one cleaves disulfide bonds? Don't remember off the top of my head, I'm sorry. If it comes up later, we'll get to it, and if we have time at the end, we'll look for it in the book. Mercaptoethanol cleaves what type of bonds? This is what you just asked. It's A, disulfide bonds. 5-dimethylamino-1-naphthalene sulfonyl chloride reacts with which functional groups? E? Yeah, it's E. It reacts with primary amines. You probably know the shortened form of that one. I don't remember it off the top of my head, but I wrote that out long ways. So, when separating subunits with 2 mercaptoethanol, the sulfhydryl subunits are alkylated with what? It's A, it's iodoacetate.
What was the reason for that again? What was the reason for that?
Ooh, they're alkylated to prevent them from reforming bonds. I believe that's why. Always, always, always double check what I say. Hydrophobic interaction chromatography does what? Ooh. B? Yeah, it's B. And nonpolar residues are what? They're hydrophobic. Okay. You see why it's named what it's named? Okay. So, gel electrophoresis can do what? Are you sure it's just A? This is, I believe I made this question just for proteins, but I believe it does everything listed. Yeah, this is for proteins. I believe I made this question for the protein review. Okay. So it's going to be D. Affinity chromatography separates molecules based on what? I need to give you guys all the rest of the answers. A, so I heard some A's that have question marks at the end. Okay, it is A. Number 37. So the peptide glytyr-arg-his-gyalin met is subjected to Edmond degradation. In the first cycle, the peptide first reacts with, phenylisothiocyanate under basic conditions. The product of this reaction is incubated with anhydrous trifluoroacetic acid and subsequently with an aqueous acid. What are the products generated in 3 cycles? C? It is going to be C. Good job. Because after you go through one full cycle of Edmond degradation, it attaches a PTH to the amino acid it cleaved. But it would not attach it to something like this, which is the only thing wrong with D, is that it attached PTH to this part. Just because it hasn't been cleaved, and it reacts specifically with ones that have been cleaved. There's probably a better explanation in the book. That's just how I understand it. Yeah, if it had said 4 cycles, it would have cleaved histidine off, and there would have been no PTH attached to gyalin and methionine. Okay. Number 38. This is another one of those questions. I'm pretty sure I didn't like this one. I don't know why I put it in there. The answer is going to be C. That's the one that I have. I wouldn't use this question to study because the pHs are really close together, and I don't exactly like that problem. I just didn't review my review. What does primary structure mean? I can't hear you guys. B? Okay, good job. It's the amino acid sequence. So, which reagent would you use to determine the number of polypeptides in a protein? What is the cheapest and fastest way to do this? It's going to be A. It'll be danzyl chloride because it cleaves every single bond of your peptide. If I'm wrong, let me know. What would you use salting out for? C? Yeah, it would be C. A protein solubility at low concentrations of the ion decreases as salt is added, which is why it's called salting out, because your substance comes out of the mixture as you add more salt. Isn't it great? Considering biochemical reactions, most enzymes that participate in chiral reactions are here's C. It is C. They're absolutely stereospecific. Which of the following functions as a regulatory protein by dephosphorylating activated enzymes? I heard an answer, and I'm not sure which one I heard. It is E. It is phosphoprotein phosphatase. Kinases phosphorylate things. Phosphatases dephosphorylate things. I don't understand your question. Okay, I can't really hear you either, so which of the following constitutes the catalytic triad? This is just a random stamp of information that you will probably need to know. C, I'm hearing C's, it is C. Okay. A small molecule that decreases the activity of an enzyme by binding to a site other than the catalytic site is termed a what? A? I have it listed as uncompetitive. It's C? Yeah. So a good transition state analog is what? C. Why would a good transition state analog bind very weakly to the enzyme? Does that make sense? Yeah, it would be A. A good transition state analog is going to bind more tightly to the enzyme than the substrate would. It's how we make some of our medications, by making transition state analogs that bind more tightly to the enzymes than its normal substrate. If you were attempting to design a new drug for the treatment of a disease by interfering with enzyme activity in the disease-causing organism, which type of inhibitor would likely be the most effective? It'd be D, because the uncompetitive inhibitor doesn't have to fall off when the substrate binds. Most enzyme-substrate interactions are transient. So if you can have an inhibitor bind to a different site than the substrate binds, you can prevent it from, completing its reaction whether or not the substrate is bound. Ready to get it? So 47 is D. In a plot of 1 over V against 1 over the concentration of S for an enzyme catalyzed reaction, the presence of a competitive inhibitor will alter the what? It's going to be B. It'll alter the intercept on the 1 over concentration of S axis. And I have a written response that's over this sort of stuff, so hopefully it'll clear it up. So 49. The number of substrate molecules converted to product in a given unit of time by a single enzyme molecule at saturation is referred to as what? It's going to be your turnover number. So number 50. The benefits of measuring the initial rate of reaction, V naught, is that the beginning of a reaction, what's true? You're just guessing. You're out? Okay. Let's see. It would be B. Changes in your concentration of substrate are negligible, so your concentration of substrate can be treated as a constant at the beginning of the reaction. Does everybody understand that? Okay. So number 51. Which of the following statements about a plot of V naught versus concentration of S for an enzyme that follows Michaelis-Menten kinetics is false? So which one just flat out doesn't make any sense at all? D? So I heard some D's. That's not correct. Well, it is correct about the type of plots, but it's not correct for this problem because it's asking what is false. So it's going to be B. At very high substrate concentrations, the velocity curve becomes a horizontal line that intersects the y-axis at Km. The steady state assumption as applied to enzyme kinetics implies what? If it's steady state, what does it mean? See, I'm getting some more C's. It is C. The enzyme substrate complex is formed and broken down at equivalent rates. For enzymes in which the slowest rate-limiting step is the reaction enzyme substrate to product, Km becomes equivalent to what? A, I heard some A's. It is A. At 54, the Lineweaver-Burk plot is used to what? It's used to solve for the rate of an enzyme reaction at infinite substrate concentrations, so it would be B. Okay. An allosteric interaction between a ligand and a protein is one in which what? What does an allosteric interaction do? It's going to be B. Yeah. Oop. I have a misspelling. It's binding, not binging. Whoops. In hemoglobin, the transition from T to R state, low to high affinity, is triggered by what? C? Yep, it's C. What does T stand for? Tense, okay, awesome. And what does R stand for? Okay, awesome job, guys. Which of the following is not correct concerning 2,3-bisphosphoglycerate? I know you guys know this one. It's also called BPG. BPG does not increase the affinity of hemoglobin for oxygen, it decreases the affinity. So it would be D. All right, we're on to number 58. Which one of the following is not among the 6 internationally accepted classes of enzymes? Did not mean to do that, sorry guys. Polymerases are not an internationally accepted class of enzyme. I believe she has a list in the PowerPoint for that chapter. And it's probably in the book as well. So the role of an enzyme in an enzyme catalyzed reaction is to what? E. Does it make the free energy, does it change the free energy change? Yeah, it doesn't change delta G. So what does your enzyme do in an enzyme catalyzed reaction? D, yes. It speeds your reaction up, or increases the rate at which substrate is converted into product. The concept of induced fit refers to the fact that induced fit is a slightly modified version of the lock and key concept of enzymes and substrates. It's a more correct way of talking about enzymes and substrate binding. So if lock and key is that one fits perfectly into another, induced fit is what? C. Substrate binding may induce a conformational change in the enzyme, which then brings catalytic groups into proper orientation. That's exactly it. Good job. Which of the following statements is true regarding collagen? We haven't looked at collagen in a long time, have we? I'm hearing some whispers of answers. What do you guys think? All right. It's going to be D. Only glycine residues can be accommodated at very tight junctions between the alpha chains, i.e. every third amino acid is glycine. Why is glycine able to be accommodated at very tight junctions? It's very small and very bendable. It doesn't have an extra R group. It's the simplest amino acid. So 62. Alpha keratin is rich in hydrophobic residues such as the following, except what? I have it down as B. I could be wrong, but that's what I found when I was looking through. But definitely go over this one yourself. But that's just what I found. Which of the following peptides would be most affected by a change in pH? So I have this one listed as A. This is where knowing your amino acids comes in, knowing their charges, knowing what they look like fully protonated and fully deprotonated. Just so you can do this on the test in case they ask something about it.
And it's most effective in a change in pH because
It's
over its charge.
Hmm?
Is it because of its charge?
Yeah, it's overall charge. Let's see if I can fit this all in one. Okay. In E. coli, the GRO-EL and GRO-ES chaperonin system assists in folding 10 to 15% of cellular proteins. Which of the following does not describe a step in the folding process of this system? I saw you answering it, and you're really close. What'd you say? No? Okay. Does anybody know? No? Okay, I'm getting a lot of blank looks. It's going to be C. Let's see. Yeah, it's going to be C. An unfolded protein is first bound to an exposed hydrophobic surface near the apical end of one GRO-EL chamber. So 65. The heme group in myoglobin binds O2. What is the function of the polypeptide? Did I say hemoglobin? I meant myoglobin. Whoops. So what do you know about the myoglobin peptide? What is it associated with? What is its prosthetic group? That's a better question. You guys should know that. Heme? Did you say heme? Yeah, it's a heme group that its prosthetic group is. So, which of these is true about that? It's going to be E, all of the above. So the Bohr effect refers to what? This is also another thing that we haven't talked about in a long time. E. It is E, yes. So the Bohr effect refers to the decrease in affinity of hemoglobin for O2 when the pH goes down. Okay. In which of the following structures is myoglobin and hemoglobin similar? Does myoglobin have a quaternary structure? No. Are their secondary structures very similar? No. No. The structure they're most similar in is their tertiary structure. A prosthetic group of a protein is a non-protein structure that what? A, yep, it's A. Prosthetic group is permanently associated with the protein. During the T to R conformational shift, iron 2 drags the F helix via a bond to the side chain of what? I heard some C's in the distance. Yeah, it's a C. So 70. Which of the following is involved in the stabilization of the prosthetic group in myoglobin? It is going to be A. Valine E11 and phenylalanine CD1. Okay. 71. In regards to heme, which of the following residues help stabilize bound O2? It's going to be B, histidine 64. I said B as in boy. I'll go bravo. In sickle cell anemia, which neutral amino acid residue in hemoglobin replaces the negatively charged glutamic acid residue? C? Yep, it is C. It's valine. So, 73. Protein dynamics is a field of study that examines the movements within a protein. Which type of protein structure determination would be most useful to study this type of charge? Or change, not charge. Oof. Does anybody know? Anybody remember from way back? Is it A? It is A. It is NMR. C, a Ramachandran plot. I don't know off the top of my head. I know I didn't put it in the review, so we'll have to go over that if there are still questions and if we have time. So, 74. Which of the following increases the affinity of hemoglobin for O2? So if the Bohr effect refers to a decrease in affinity during a decrease in pH, what's going to increase the affinity? D? Yeah, it's going to be D as in dog. An increase in pH is going to increase your affinity for oxygen. So, 75. The interactions of ligands with proteins are what? Which question? It is D. They're usually transient. '76. Which of the following statements about protein ligand binding is correct? Is it C? Why would your Ka, a large Ka, mean a weak affinity? Is it what? E as in echo? Yeah, it's going to be E. The larger the Ka, the smaller the Kd. All right. So, We are on to the written response portions. I know that your final is only going to be multiple choice. However, I am going over concepts that you will probably need to know for your final. Okay. Up. Yeah. And did you? Yeah, you got your charger and everything. Okay, awesome. All right, thanks for coming. Have a good day. All right. So, create an 8 nucleotide reverse primer for the underlying portion of DNA. Where would you even start with this problem? Yeah, you'd start with the complementary strand. So where would you start? Would you start your complementary strand here or here for a reverse primer? The last one? So we'd start here. And we're going to go 8 nucleotides. So 1, 2, 3, 4, 5, 6, 7, 8. So we would stop here. So these are the nucleotides that we are translating into a primer. Does anybody know what that primer is going to read? Is it going to read 5 to 3 or 3 to 5? 5 to 3? Are you sure? So you're saying your 5' is going to be there?
Okay, so the 5' for the primer
Should be here, yes. Yeah, that's what you're saying? Okay, good job. So, and then it's going to be 3'. So, what is your primer going to be? So, I can't really hear you guys, so it's going to be from the 3' end, it's going to be CC, T, A, T, T, A, G. Everybody get that? Okay. So that's pretty simple. Now how do we write it from 5' to 3'? It's going to be G, A, T, T, A, T, C, C. Okay? They can mess you up on multiple choice by doing that. Just be sure to always look at how they're starting it. All right.
Forward primer would just be the same as like the first 8, right?
I want to say yes, but I don't know for certain. Be sure to double check and do your own research. I couldn't find anything like solid about it and when I was trying to find it, so it's up in the air. All right, so membrane potentials. In this equation, what is F? Okay, so it's going to be Faraday's constant, and we use the one, let me find it on my paper, we use the one that is 96.5 kilojoules per mole. So, and if R is in moles, are you going to have to do some unit conversions? Yeah, you're going to have to do some unit conversions for these, especially with millivolts, micromolar, and millimolar, all that fun stuff. So, what does T mean? Like, what unit are we using for T? Kelvin? Okay, awesome. What is Z? Yeah, it's the ionic charge of the ion that you're trying to find. And then, does it have any units? No, it has no units. So if we're using potassium, what is our Z going to be? So our Z is going to be 1. All right. And then my micromolar is Okay. Just trying to get all the unit conversions done. All right, so can anybody tell me how to set this equation up? And what else we need to do to make it so that you can have all of your units either cancel out or make sense? Okay. Yeah, you just plug it into the equation. Which one did I forget to change the units for? Okay. So what is delta G's normal units? I list it as kilojoules per mole. And I mean, you can just convert back to kilojoules because that's how I did it. I converted Faraday's constant into joules per mole, and then I reconverted it into kilojoules per mole. So we can do it either way. We'll just use Faraday's again. It's going to be, where did I have that? It's going to be okay. So now we set up our equation. R is 8.314. Units are very important. Always remember to write your units down just in case stuff doesn't work out, so you can always go back and find out where you messed up. Times. So what would our temperature in Kelvin be if it's at 30 degrees C? 30 what? 302. So how do you translate? Yeah, 303. It's 273 that you add to your Celsius. Times the. So if you're going against a concentration gradient and the higher concentration is within the cell, which one would you put here? Which concentration would you put as inside the cell? Which one of these is a higher concentration? I don't understand. Yeah, if the higher concentration is within the cell of the potassium ions, which number would you use as concentration of S inside cell? So if this is 9 times 10 to the negative 6 molar, and that one is 0.35 molar, which one's a higher concentration? 350? Yeah, so 0.35 molar is the higher concentration. It's within the cell. So then you would divide it by 9 times 10 to the negative 6 molar. Okay. And then you would add your Z, which we figured out was 1, times your Faraday's, times your membrane potential. Okay, does that look good to everybody? All right. And then you just divide that whole thing by 1,000 to get kilojoules, to get delta G in kilojoules per mole. Does that make sense? So, delta G is equal to what in kilojoules per mole? So when I did this, I got about 36 kilojoules per mole. I'm not saying I'm the best at these. Definitely recheck and do it yourself. Make sure that you know how to do these. I just wanted to give you guys some examples because this will definitely be on your final. Okay? Do we want to do the second one, or do you guys think you're okay? Do the second one? Okay. So, it's the same type thing. We're going to translate all of our units into the normal units. So our micromolar and our millimolar are going to get converted into molar, millivolts to volts, Celsius to Kelvin. And I think Z is going to stay the same because we're still using potassium ions. Does that make sense to everybody? Okay. What? Since we're still using potassium ions between the two, yeah, Z is going to stay the same because potassium always has a charge of +1. So if you have something with a charge, yes?
For the pH, is it just above?
What?
Is it just above for the pH?
The pH I just put in there because I forgot that pH actually relates to membrane potential. If you want to, you can change this to 7.0, or you can just cancel it out completely because you don't actually need pH in these types of problems for how much information I gave you. But if you're not given membrane potential, you can find membrane potential if you're given the pH outside and inside the cell. Okay? So this does not matter. That does not matter.
So we have to be given 2 pHs?
I believe so. Hashim didn't go over one where you had 2 pHs, so I'm not entirely certain. So definitely look through your book, see if you can find it, or go talk to the professors. Okay. So that's going to be okay. So trying to do math in my head is not going well today. 311 Kelvin. Okay. Times the oh, I'm running out of room. Whoops. Plus 1 times. Okay, does that look right to everybody? Everybody like how I inputted things? It's a formula, so hopefully you're just going to be given plug and chug stuff. Okay. So this is again going against the concentration gradient. So what are we expecting to get for our delta G? Are we expecting positive or negative? Positive, okay. So does that mean it's going to be spontaneous or non-spontaneous? Okay, and in order to have this reaction move forward, what are we going to pair it with? Okay, an exergonic reaction, what is the energy currency of the cell? ATP, yeah. So would you pair it with ATP hydrolysis? Most likely? Yes, okay. Awesome job, guys. So for this one, I found that delta G equaled about 31 kilojoules per mole. Again, do it yourself, figure it out. I'm not saying I know everything about this.
Can you go over why we put 0.28 on
So what are you talking about?
Why do we put the 0.28 above?
0.28 for both. Where are you looking?
The 0.28.
Right here? Because if you're moving, if the higher concentration is within the cell, and when you take the ln of the solute within the cell divided by the solute outside the cell, and the higher concentration is within the cell, you'd put the higher number with the concentration of, substrate within the cell. Yeah. Does that make more sense? Or?
Is it always the larger number?
It depends on what the question asks you, because the higher concentration can be outside of the cell. Usually with waste products, your cell will pump them outside and it'll be a higher concentration outside. Just look at the problem, make sure you understand it, ask questions if you need to, that sort of thing.
So what's the keyword for that question? What's the keyword for that?
I actually didn't put the keyword within my question by accident. Both of these are supposed to say that the higher concentration is within the cell. Okay? So if I was saying that it was taking it into the cell against its concentration gradient, you would say that the higher concentration is within the cell. Okay? Everybody good? Okay. Okay. So, back to amino acids. What's the 3-letter sequence of this peptide? If you printed this out the day I posted it, you would have gotten the answer printed out as well. I changed it when I realized that I did that. So, what's this guy? A? Hmm?
Is that I?
This is alanine, right? So that would be A, right? What would this guy be? Who is this guy? Lysine? Isoleucine, or is it leucine? It's leucine? Okay, so what is leucine's 1-letter abbreviation? L? Okay. Isoleucine. This is leucine. This is not isoleucine. What does isoleucine look like? Isoleucine, instead of having the V here, you wouldn't have one of these, and you would have a methyl group coming off of this one. Okay? So A, L. What is this one? Lysine. What is lysine's single-letter abbreviation? K. Okay. K. What is this guy? Okay. What is its 1-letter abbreviation? D. This one? Phenylalanine. What is this one? F. Awesome. And then this one? Does it have any R group? Glycine. Okay, what's its 1 letter? G. Okay. And then we've already seen this guy. Alanine. Okay. This one? Proline. Isn't proline fun looking? What's its single letter abbreviation? P. Okay. What's this one? We've seen this one already too. Glycine. Okay. And then what's this one? Leucine again. Awesome. All right. So does everybody know the 3-letter abbreviations for this? Yeah? Do we need to go over it? No? Okay. What does trypsin do? If you know what it does, you can react it with this peptide to make its fragments. Getting lots of blank stares. Trypsin cleaves after lysine and arginine. Okay, except when what? It's followed by a proline. Okay, so lysine and arginine. I believe that's arginine, or is it? Yeah, arginine. Yeah. Okay, so what would the fragments be if you react it with trypsin? Okay. And what? Yeah, and then the rest of it, exactly. D, F, G, A, P, G, L. Okay. What does chymotrypsin do? Yeah, it cleaves after the C-terminus, or cleaves the C-terminus of phenylalanine, tyrosine, and tryptophan, aka hydrophobic residues. Woo. Okay. And do we have any cleavages here? It cleaves leucine slowly. I'm not sure if they'll ask you something like that, but if they do, definitely know that. So if it also cleaves leucine slowly. Okay. So how many cleavages would we have in this peptide? 1? Okay. And I'm sure if they ask you something like this, they'll let you know if they let it react for a long time or if they only let it react for a short time about the leucine thing. Okay? So you'd have 1 cleavage. What would the cleavage be? After the phenylalanine. Awesome. So D. Okay. What about cyanogen bromide? What does that do? It cleaves the C-terminus of methionine residues. Okay. Do we have any cleavages here? No. So if you're asked something like that, you would just write down the whole peptide sequence again. Okay, everybody got it? Awesome. So if you have 1 round of Edmond degradation, what would be cleaved? So it would cleave this very first one, right here. That happy little guy. Okay? And then let me fill this out. ALKD. Okay. Okay. And then which amino acids would fluoresce when treated with danzyl chloride? What does danzyl chloride like? Okay, so which ones would it cleave and fluoresce? Or and attach a fluorescing to? Lysine, okay. So lysine would be 1. Okay, what would be the other? This one? Yeah. So, does everybody like that? Okay. Now we're on to number 4 of the written responses. So a peptide sequence was fragmented using 2 different methods to generate the following sequences. Determine the sequence of the peptide. You guys remember how to do that, right? Okay, so you know it starts with VM. Does everybody agree with that? I know I do. So VM. What did you say? Awesome. Okay. Good job. So, which enzyme or chemical was used to generate the fragments in method 1? Trypsin? Does everybody like trypsin? It would be trypsin. We did just go over this. So which enzyme or chemical was used to generate the fragments in method 2? Cyanogen bromide. Does everybody like that one? Okay, awesome. All right, any questions, comments, concerns about that one? That's where cyanogen bromide cleaves. Cyanogen bromide cleaves on the C-terminus of methionine residues. Just so. Okay. So everybody was good with that one? Awesome. That was awful. All right. So, a 10 nucleotide long DNA template was sequenced using the chain termination method, aka the Sanger method. The template was flanked with a 3 nucleotide primer and reacted with DNA polymerase and various dNTPs and ddNTPs, and the resulting fragments were analyzed by gel electrophoresis. Given the sequence of the template strand, determine which dNTPs and ddNTPs were used in each of the 5 lanes. So, let's start with our primer. What's our primer? I heard it, but I don't know who said it. So what's our primer going to be? If the template is AAA, what is your complementary? Okay, good job, guys. All right. So how do we go about figuring this out? Because we know our entire template strand, so does that mean we know our entire complementary strand? Yes, okay. So you would fill this out as C, G, C, A, G, T, G, T, T, T. Does that look right to everybody? And then you would write your primer below your gel, because your primer is going to be attached to every single piece of sample that you have. So it's not going to influence how far it moves down the gel. Okay? Everybody understand that? Okay. So then we would write G, T, G, A, C, G, C. Does that look good to everybody? Yes, no, maybe so? Okay. So in sample 1, that happy little guy right there, which DNTPs and which DDNTPs are within that sample? Does it go to completion? Meaning, do you have a line all the way up at the top? Yes, okay, so does that mean it has all of its deoxynucleotides? Yes, okay. So it has DATP, DTTP, DGTP, DCTP, and where does it always have a line? So it always has a line at G. So which dideoxy does it have? I heard it, but I don't know who said it. It would be DDGTP. Okay? And then you kind of go through that deductive reasoning for all of them. So what about in sample 2? So does it have all of its DNTPs? Okay. Does it have any DDNTPs? No. Okay, awesome. So this has DATP, DTTP, DGTP, DCTP, and no DDNTPs. Okay? Everybody understand that? All right. What about C in sample 3? Does it go all the way to completion? No. Okay, so what does that tell you about this sample? Yeah, so it's missing a DNTP. Which DNTP is it missing? Like the one you know for sure it's missing? So, but you have a G here, and then you have another line at G there. So does it? Which one? So it doesn't have a line all the way up here. So that means it has to be missing. Yeah, but if it goes past this G, that means it has to have at least some of its dioxy NTP or GTP. So if it would it make a line at the last G if it's missing a DATP? Would it be able to continue if there's no DATP?
Oh, I see.
Yeah. So it's missing its DATP, and it may or may not have DCTP. You don't know that. And so it has DTTP, DGTP, and those are the two we know for certain. And we also know it has what dideoxy? So it has DDGTP as well. And those are the three you know for certain it has. It may or may not have DCTP. Okay? So for sample 4, what's present and what's not present? So all of them are present? Okay. There we go. Okay. Does everybody agree? So it does have DDCTP. Does it have anything else? We already know it has all of its DNTPs. Does it have any other dideoxys? No? Awesome. So, for sample 5, what DNTPs are present? So, DGTP is present. I heard that somewhere. All right. Is there a DTTP present, though? Well, if you have DGTP and you also have a DTTP, this could go on further, at least one more further.
Oh, I thought you would need the GTP for it to go on. No?
No, this line here means that there is DDTTP in there.
Yeah.
And if it doesn't go any further, when you already know it has DGTP, you know that it cannot have any DTTP. Right? Does everybody understand that? Okay. So, and is there any way to know if it has DATP or DCTP? No. So it may or may not have it, but you know for certain that those two are what it's got inside of it, right? Okay. Is everybody okay with that? Awesome. All right. All right, I'll just zoom in on this. Okay. So, I'm going to use all my different colors because this graph can get confusing if you don't use different colors. So, without an inhibitor, where is Vmax? We'll just put it at 80, just because it looks like it's still going to go up a little bit more from where it is right now. And that makes our 1/2 Vmax a little bit easier for that one. And which shares a Vmax with, which inhibitor shares a Vmax with the without inhibitor line? So there's 3 different types of inhibitors, and one of them shares the Vmax of the enzyme without an inhibitor. Which one is it? I know there's a table in your PowerPoint somewhere. Your competitive inhibitor shares a Vmax with the enzyme without an inhibitor. Okay. So what does that mean? Is it going to share a 1/2 Vmax with the enzyme without an inhibitor? If they share the same Vmax, are they going to share the same 1/2 Vmax? I'm getting nods, yeah, absolutely. So since we placed it at 80, our 1/2 Vmax is going to be at 40. Okay. All right. So how do you find your Km? Your Km is where your 1/2 Vmax intersects your line. So here's your enzyme without an inhibitor. So its Km is going to end up here. That's the wrong one. All right. So where is your competitive inhibitor Km going to land? Just follow this line, and then you hit it at 1/2 Vmax, and you go down. And there is your Km for your competitive inhibitor. All right? Everybody got that? So your non-competitive Vmax, which you can see, that's non-competitive. Does non-competitive inhibitors change the Vmax? They do. Non-competitive inhibitors change the Vmax. So we'll put that slightly above the C line. So if it's at about 38, oh geez, I'm really bad at math. 16, I think. It'd be about 16 is your 1/2 Vmax. Or 18. Okay. So where's your Km for non-competitive inhibitors? Does your Km for non-competitive inhibitors change from the one that doesn't have an inhibitor, from the enzyme that doesn't have an inhibitor? Yes, no, maybe so? Yes, okay. Are you sure about that, though? All right. So your Km for your non-competitive inhibitor should be the same as the enzyme without an inhibitor. Okay, so oh wait, am I thinking about the wrong one?
Km never stays the same for any. It always changes no matter what inhibitor you're using.
Okay. Is that what the book says? Okay. Then I found the wrong thing then. Okay. So where would Km be for this? I think it would end up somewhere around here. Km. All right. And then what about uncompetitive? What does uncompetitive change? It changes Km, okay. And Vmax? All right, awesome. So would your Vmax be higher or lower? Awesome. And what does it do to your Km? Okay. So we'll have there we go. So does that look about right to everybody? Where would your Km end up for the D line? If following the line, our Km ends up here, but I believe it just decreases it, so. All right, is everybody okay with that graph? Okay. Now we're on to our Lineweaver-Burk plots. What's A? Competitive? Okay, awesome. So it is competitive. Where is 1/2 Vmax, or 1/Vmax supposed to be located? I heard the y-intercept, and that's exactly where it's supposed to be. So right here is the 1/Vmax. Okay. And what are these intercepts called on the x-axis? So it's -1/alpha Km. And where are your alpha numbers? Where can you find them? So if there's no inhibitor here, alpha is equal to 1. And then as you go up, your inhibitor concentration also increases. So you can say that this line is alpha 2, this line is alpha 4, and this line is alpha 6. So, and then for this -1 over alpha Km, you just insert your alpha into that alpha place. Everybody understand that? Okay. And then what is the slope for competitive inhibitors? Okay. So the slope for competitive inhibitors is going to be alpha times Km divided by Vmax. Everybody got that? Okay. What about B? What type of inhibitor is that? Uncompetitive? Okay, awesome. All right. So where is alpha prime over Vmax located? Is it your x or your y-intercept? Alpha prime over Vmax is your x or y-intercept? Okay, awesome. It is your y-intercept. All right. So what is your x-intercept called? So negative alpha prime over Km. What is your slope of uncompetitive inhibitors? So the slope of uncompetitive inhibitors is Km divided by Vmax. And you can tell this because they're all parallel to each other. They never touch. Okay. And the last one, what is that? Mixed. It's also called what? Awesome. Did I misspell that? All right. So what is your y-intercept called? Yeah, it's alpha prime over Vmax. All right, what's your x-intercept called? It's negative alpha prime over alpha Km. I forgot to give you alpha prime numbers. We'll go back to that one. So, for this line, alpha equals alpha prime, which is 1. That's without any inhibitor. For this one, you can say that alpha is equal to 1.5 and alpha prime is equal to 1.25. These are pretty much arbitrary numbers, but you just have to know that they are different. Okay? And then we have alpha is equal to 2, and alpha prime would be equal to 1.5. And then for the last line, alpha is equal to 2.5, and alpha prime is equal to 2.25. Okay? All right, so what is the slope of mixed or non-competitive inhibitors? It's alpha times Km over Vmax. All right? And then let's go back to uncompetitive and label those numbers as well, or those alpha numbers. So do you only use alpha prime for uncompetitive inhibitors? Do you see any alphas here or just alpha primes? Just alpha primes. So do you need to worry about alpha numbers? No. Okay. So without an inhibitor, what's your alpha prime going to be? 1. Awesome. So the alpha prime of line 2 is just going to be 1.5. Alpha prime of line 3 is just going to be 2. And alpha prime of the final line is going to be 2.5. All right. And that should be the end of the review. Do we have any extra questions or comments, concerns? Still got a few minutes. Stopped about 15 minutes early. And I'm going to go ahead and pass out these, what are they called? Evaluations. Yeah, you can come up and take a picture of this one too. This one has a little bit more information if you need it. So. No, you didn't do one of these. So, you're welcome. Thank you for coming. Make sure to clean up after yourselves and all that awesome stuff. If you guys would please fill out the evaluation.